Thanks, Anna. That was excellent talk. I really enjoyed that. And so we've got a bit of time for some questions right now. If anyone has a burning question for Anna. I can't see anything in the chat right now. I'll ask you one while we're waiting. So the data set that you used, and you're saying the sort of the association, the GDP between different countries, do you think that might be biased by the kind of, what we're looking at and what we have at the moment? Is there a way in which this should be improved?
Yeah, absolutely. I mean, there's a high prevalence of isolates from UK and US in Papua New Guinea. It's just, the way sequencing is at the moment. I think there is a bit of more of a push to get a bit more sequences from, for example, LMICs. But I think the association is there. There is a high prevalence of plasmids in LMIC countries. I think there's no doubt about that. But yeah, I agree. We need more sequences from less wealthy countries.
And it's undoubtedly due to antibiotic usage as well, really, and that sort of.
I think so, yeah. Yeah, this might be a differing antimicrobial use between countries.
Yeah. So there's a couple of questions that come through. Sylvia Brewston, thanks for this interesting talk. What do you actually mean with lineages?
So lineages were defined with the core genome MLST. Odile is much better placed probably to answer this question. So it was done in a collaboration with Odile, and we used core genome MLST scheme of 400. So basically every lineage would cluster isolates that have 400 or less loci, similar loci. So
yeah, they're clusters of related isolates that differ from each other in 400 or fewer loci. But as I said in my talk, you can use different thresholds as well if you want to. And we're currently working to try to have a bit more stability in the sort of core genome cluster nomenclature that is used, so that ultimately we can have something which is reproducible over time as well, and a structure that remains stable, really, and consistent. Yvonne, I see you put your camera on and you've asked a question. Do you want me to read it out for you, or do you want to say it? Yeah. So great talk. Thank you. Is there actually anything known about the influence of copy number on AMR?
Yeah, good question. This is something we're actually looking at the moment. So there are some data from other labs that show that, for example, at least for beta-lactamase plasmid, I can't say anything about the conjugated plasmid because we don't know anything about that. But beta-lactamase plasmid apparently is in 4 copies, and it can have. So there is more. So TEM135, for example, has higher penicillin resistance, but that's irrespective of the copy number. So currently, the data from other labs show no, but we are looking at it.
Yeah, I thought that, Sylvia, thank you for that question. Just to come back on that about the lineage question, I think that's a very good question. We might want to pick it up a bit later, actually. So obviously we're seeing this with COVID, but this is true with everything. It's the importance of international nomenclatures, and the ST nomenclatures are relatively straightforward. Defining lineages, of course, is very complicated for organisms such as the gonococcus that recombine so much. Maybe a good opportunity at this forum to think a little bit about that, because as Odile says, we have to come up with stable nomenclature to have broad agreement. And we've had for many years a committee for Neisseria MLST, which was mostly meningococcal, but I just wonder whether we should think about getting together a group of people to advise on what the names of lineages should be and what their cutoff should be, because naming is very important. It's also not trivial, actually. Even in COVID, they have their problems, and that's relatively straightforward because it's wholly it's wholly mutational and completely clonal. But with the gonococcus, it's more difficult. So I think that's a really good question, and maybe something we should take on as an action point, because the important point is that we do have stable nomenclatures that everybody buys into and uses. And clearly, the interesting thing is that there is structure there at this whole genome level that we need to represent in one way or another. So maybe we can pick that up at the general discussion at the end, because I'm sure there might be others who might want to contribute to that.
Thank you. Actually, I just indeed want to add that it will be more difficult for Neisseria gonorrhea, which is very polymorphic. But if these plasmids are there or they are not there, so you need 2 names or more names.
Yeah. Yes, it's
Thank
not
you.
trivial, unfortunately, but it
No.
is very important.
Okay.
Sorry, shall I mute? Have you got some more questions? I know there's a couple more in the chat. I'm happy to take
Let's just take those quickly and then move. Yeah, yeah.
Okay, great. So let me see. So Bill's before you. Sorry, Hank. Is it known if acquisition of a beta-lactamase plasmid confers a fitness defect on the host GC strain?
Yeah, again, I can't answer that just yet, because that's exactly what we're doing at this very moment, and we were a bit delayed with the whole pandemic and homeschooling. But we don't think there is much fitness costs, but we don't know. We also don't know whether there's any compensatory mutations on the chromosome once the plasmid is acquired. So yeah.
Excellent, thank you. Hank, do you want to ask your question?
Yeah, hi Anna. So the cryptic plasmid, I have to bring it up to people in my lab, and that was the first sequence ever, first thing ever sequenced from Neisseria gonorrhea by the Swedes. I know you're focused on antibiotic resistance, but is any of your analysis going to reveal functions of the cryptic plasmid and make it less cryptic?
Yeah, I think we should actually. So it's called cryptic because it's not associated with the phenotype, but there were some, if you sort of delve into sort of like microarray data in sort of like, you know, growing Neisseria gonorrhea in sort of different conditions, you actually see in those data that some sort of genes are actually upregulated on the cryptic plasmid. So I'm sure there is a phenotype associated with it. We have also found a very small number of isolates that don't have it. But they have only 2 genes belonging to the cryptic plasmid on the chromosome, and that was a TA system, and it's associated with a type 4 with a different type 4 secretion system, so a third type 4 secretion system for Neisseria gonorrhea. So certainly there is something going on, but yeah, we need to sort of like put a little bit more focus on it.
And thank you, Anna and Hank. So one final question from you, Linda. Do you think the beta-lactamase on the plasmid is a real threat for resistance against ceftriaxone, because the prevalence of this plasmid is so low in high-income countries where ceftriaxone is more often used?
Yeah, so I do think there is probably just a matter of time before basically that one mutation arises that turns into an ESBL. I think probably it will happen probably in LMICs, so in less wealthy countries where there is a high prevalence of plasmid, we're probably more likely to see it. For our data show that in high-income countries, the resistance against ceftriaxone and cefixime are more chromosomally mediated. And that may be also due to sort of like differing lineages between the two sort of sets of countries. Yeah.
Excellent. Okay, thank you very much. We shall carry on with our next talk. Thanks, Anna.